Yes, dear students, today we shall start Bio-Inorganic Chemistry. So actually you are all belonging to biology stream, so this chapter may be somewhat useful to you. And here generally we are expecting short answers only, not an essay question. So what is this bio-inorganic chemistry? So bio-inorganic chemistry is a branch of chemistry that deals with role of metals and non-metals involved in biological processes. So biological processes. Biological processes means the chemical reactions that are involved in living systems. So living systems include plants and animals. Plants and animals. So for plants and animals we are using alternative terms, that is flora for plants and fauna for animals. Flora and fauna, these are the two terms used for plants and animals. So bio-inorganic chemistry, or it is also called inorganic biochemistry, which deals with, it is a branch of chemistry that deals with the study of role of metals as well as non-metals which are involved in biological processes. Now what is the main concern of this bio-inorganic chemistry? The first aspect is the biological functions of different elements. The biological functions of different elements which are actually known to form inorganic compounds. So such elements we are studying and which are playing a vital role in biological systems. And secondly, transport of ions and its storage. Transport of ions as well as its storage in living systems. Then thirdly, it also deals with toxicity. So many metals or non-metals are generally elements. Many elements, they are toxic in their nature, so it deals with, it is mainly concerned with the toxicity of these elements and their compounds also, and what are the methods we are using for overcoming or minimizing this toxicity. And next, fourth one, we have the atmospheric nitrogen. Artificial fixation of atmospheric nitrogen, that is also one of the important aspects of this bio-inorganic chemistry. Now here already you have learned in leguminaceae, legumes, their root nodules, they are having rhizobium, and we have blue, green algae. There you have learned nostoc and anamina. So these are the things which are having the ability to fix the atmospheric nitrogen directly into the nitrogen compounds. So this comes under artificial fixation of atmospheric nitrogen. And next to fifth we have Yes. So here we have seen that the bio-inorganic chemistry is mainly concerned with the biological functions of different elements which are known to form many inorganic compounds generally. Secondly, we have transport of ions and its storage in living systems. Then toxicity of these elements and their compounds, and what are the methods we are using to minimize that toxicity. Then we have artificial nitrogen fixation, that is the another one. And the metals, many metals, they are used in the drugs, and also metals are used as probes. Probes means used for investigation purpose. So you might have seen metal detector. So that is identifying what are the metals that person is carrying. So like that, here some of the metals, they are used in the preparation of drugs, and also they are used as probes also. And sixth one, this is very important point, that is we can extract metals. So extraction of metals from ores by using microorganisms. So in the microorganisms we are using either bacteria, or we may use archaea. I think you might have heard about this archaea. So here, actually this one, it is the intermediate that is present between bacteria and eukaryotes. Eukaryotes means these are the living organisms which are having a distinct cell with a distinct nucleus, and DNA is present in the form of the chromosomes. So archaea is a connecting link or intermediate between bacteria and eukaryotes. So you may get this, that is what is the main concern or importance or significance of this bio-inorganic chemistry. Then you have to write these 6 points. Okay, so this is one. Next we shall see the classification of elements which are involved in biological systems. So far, in the living organisms, that is both plants and animals, nearly 30 elements were identified. They are performing their role in biological reactions, biological systems. So these 30 elements, they are classified into 2 categories. One is essential and another one is non-essential. Essential elements and non-essential elements. Non-essential elements. So here, these essential elements, actually they cannot be synthesized. They can't be synthesized in the body. Whereas non-essential elements, they can be synthesized in the body. So, like this here it is elements, 30 elements, they are classified into 2 categories, that is essential and non-essential elements. These elements, they cannot be synthesized either in plants or in animals. And non-essential elements, they can be prepared in the body. Now, these essential elements, they are once again classified into 3 categories. 3 categories. Based on what? Based on the amount of that particular element. So here we are using weight percent. What is the weight percent? Weight percent is nothing but the weight of an element, and that element is present in the form of a compound. So weight of that compound into 100. This is the equation we are using. So weight of the element by weight of the compound into 100, or we can also find out like this: weight percent is equal to weight of solute by weight of solution into 100. Okay, so like that here elements are, these essential elements, they are once again classified into three categories based on their weight percent. Suppose if the elements are having greater than 1.0 weight percent, then they are called major or bulk elements. Greater than 1 weight percent. Suppose if the weight percent is 0.1 to 1 weight percent, then it is called, for major we use opposite word minor, and bulk it will be, here we write macro. So 0.1 to 1 weight percent, and last one is trace elements. We have trace elements whose weight percent is less than 0.1 weight percent. So like this, based on the weight percent, it is classified into, the essential elements are classified into three categories, that is the bulk elements, then macro elements, then last one is trace elements. And you can go through the elements. I have already given the notes. You just check up which elements fall under which category. Then I will go to the next aspect, that is geochemical effect on distribution of elements. So geochemical effect on distribution of elements. Or it is also called migration of elements. Say for example, in our Kannur district we have Jonnagiri. There we find diamonds. And suppose if you go to Bangalore, Karnataka, there we have KGF, Kolar gold fields. So here, at some places, some elements are present in greater concentration. The concentration is more. They are present in the bulk amounts. And at other places, its concentration or its amount is very, very less. So why these elements, they are distributed? That is unequal distribution of these elements. Why it is happening? What is that geochemical effect that is influencing this distribution of elements? So migration of elements or distribution of elements which is influenced by this geochemical effect. Now here let us know what is this geochemistry. So according to geochemistry, the chemistry that deals with Earth's crust and oceans. So geochemistry, according to this branch, Earth's crust and its oceans they are the major geochemical systems. Major geochemical systems. So we have two major geochemical systems. One is Earth's crust and another one is oceans. Now this Earth's crust, it consists of discrete means separate. So discrete spheres. One is rocks, another one is fluids, Next we have gases, and finally we have biology, that is living organisms. So Earth's crust, it is classified into, or it is consisting of discrete spheres that includes mainly rocks, fluids, gases and biology. What are fluids? What is the meaning of a fluid? Literary meaning is they tend to flow. They will flow from one place to the another place. So generally gases and liquids, they are combiningly called as fluids. That we know it. So here between these geochemical systems, both matter and energy will be exchanged. So generally matter and energy, matter includes elements, compounds, etc. So matter as well as energy, it is being exchanged between any two geochemical systems. How long this process will take place? This process will take place until they are evenly distributed and attain equilibrium. That means the concentrations must be equal on both sides. So till then it is occurring. For that it is taking how much time? So it is taking so many thousands of years. That means we have to consider both time and space also. Space means from one region to the another region. So like that here, between any two geochemical systems, the matter as well as energy, it is being exchanged as per the time and space until they are evenly distributed and attain the equilibrium. Okay? Now we shall learn why these elements are unequally distributed. For that, what is the main reason? The main reason is concentration gradient. This you have to understand here. What is this concentration gradient? The difference in concentration of matter between any two geochemical spheres. Any two geochemical spheres with respect to time and space. So here the difference in concentration of matter, or we can say particles. So due to the difference in the concentration of particles or matter between any two regions, that is called as a concentration gradient. So here this geochemical effect in the form of concentration gradient is responsible for the distribution of, which distribution? Unequal distribution of the elements. So this is the migration of the elements also. So concentration gradient is the one of the prominent factor that is influencing this distribution of elements in the Earth's crust. And what are the other factors that influence this distribution of elements? So this is number one, and secondly we have thermal equilibrium of Earth. Then we have this should be read as pi, Phi. So that is nothing but ionic potential. So ionic potential, it is a ratio, ionic potential, it is a ratio of charge and radius. So the other factor that is influencing this distribution of elements is ionic potential. Then we have temperature also separately. Then fifth one, tectonic and magnetic movements. So magnetic movements, tectonic movements, they are also responsible for distribution of these elements in the Earth's crust. Then we have sixth one, bio-mining. What is this bio-mining? Or it is also called bio-leaching. So bio-mining or bio-leaching means nothing but, already I have mentioned, Bio-Inorganic Chemistry last point, that is the process of extraction of metals from ores by using microorganisms like bacteria and archaea. That process is called as the bio-mining, or it is also called bio-leaching. Okay, and the last and final term, that is metasomatosis. What is this metasomatosis? It is the process of change of composition and texture. Say for example, if you take a cloth, it is having the fibers. So how these fibers are arranged, that constitutes the texture. So here the process of changing of both composition and texture of a rock, that is called as metasomatosis. And last and final, eighth one is solution effect. What this solution effect means? Generally, it was assumed that 1 kg of sea water, if you consider 1 kg of water, generally 600 grams of rock will pass into the solution. So that will be converted into a solution. So that is called as the solution effect. So in the geochemical effect on the distribution of elements, the main predominant factor is concentration gradient. What is this concentration gradient? It is the difference in concentrations of the particles or the matter between any two geochemical spheres. Whether it is rocks or fluids, or it is rods or biology, whatever it may be. We have 4 spheres in between any two that exchange will occur. Okay? Then, next aspect is in the case of cells, we find extracellular fluids and intracellular fluids. So the fluid that is present inside the cell, fluid that is present outside the cell. Here, in the case of physiology, what this physiology says? The biological processes or the chemical reactions that are occurring in living organisms, that constitutes this physiology. So in physiology, we have mainly 2 gradients. One is concentration gradient, and secondly we have potential gradient. Now we shall understand what is the meaning of this concentration gradient and potential gradient. Already I have explained this. Concentration gradient means it is nothing but the difference in the concentration of ions. So here In the extracellular fluids, we have some ions. What are they? We will shortly learn. And in the intracellular fluids also we find many ions. So actually I have written these two words in connection with, we are going to learn sodium potassium pump. Sodium potassium pump, that is a very important either essay or short answer. So for understanding this, the mechanism of this sodium potassium pump, one must have these terms in mind. One is a concentration gradient and another one is a potential gradient. Now here, another important word we need is, let me finish this one first. Gradient. Actually, literary meaning, gradient means it is a gradual change. A gradual change. That is called as a gradient. So this gradual change may be either it is an increase or it may be a decrease. Okay? So gradual change includes either increase or a decrease with respect to distance. So the elements distribution, it is changing with the distance in the Earth's crust. So either increase or decrease, that is called as a gradual change. That is nothing but the gradient. So here concentration gradient means the difference in the concentration of ions. Okay, now here, say for example, sodium it is having one positive charge, potassium one positive because they come under first year group elements, and calcium it is having +2. So like that here, these are the sum of the ions which are carrying the charge. So now the second term, potential gradient, is connected with the charge. So for expressing the flow of charge, generally we use regarding the charge, we use the term electric potential. What is this potential difference? Now, let us take one example first. Let me clarify first. Now I have taken here a magnet. Now, this magnet, it will have some amount of space around it. It can show its influence. Say for example, if I bring a nail within this boundary line, definitely this magnet will attract this one. So the space around a magnet where its influence is felt is called as the magnetic field. I will repeat. The space around a magnet where its influence is felt. Suppose if we bring iron pieces, definitely it will attract. Okay. Now this is called as magnetic field. Now similarly, suppose if I take a charged body. Now if another charged body, say for example, if I bring a positive charge within this boundary line, there will be a repulsion. Suppose if I bring a negatively charged body, then there will be an attraction. So this is called electric field. What is electric field? It's a definition. The space around a charged body where its influence is felt is called as the electric field. So if you understand this one, then we can easily understand what is this electric potential and potential difference. Now, it is the amount of work done. Amount of work done in moving a unit positive charge from infinity to that point, a particular point in the electric field is called electric potential. I will repeat. The amount of work done in moving a unit positive charge. So we are taking that as the standard, unit positive charge. is taken as the standard here. For moving that unit positive charge from infinity to that point in the electric field is called as the electric potential. Now what is the potential difference? Its definition is the amount of work done in moving a unit positive charge from A to B. That means some two fixed points. That is called as the potential difference. It is expressed in volts, etc., as I don't want. Now here we must understand what is this potential gradient. Now in the case of cellular fluids, so cellular fluids are two types, okay, that is extracellular and intracellular. So in the cellular fluids generally concentration gradient is existing. Why? Because unequal distribution. So unequal distribution of ions, that is responsible for that concentration gradient. Now, say for example, I am citing here sodium positive, potassium positive, calcium positive, bi-positive ion. So, like that here, if ions are unequally distributed, then these ions are carrying charges. So, there is a difference in the amount of charge that is distributed in the cellular fluids. That is expressed in terms of potential. So, the difference in potentials of the ions. So, ions charges are generally expressed in terms of potential only. Okay, that's why we have used here the term potential. Gradient means a change. That means concentration is different. The charge concentration is different. That is called the potential gradient. Now, I think you might have understood that concentration gradient and the second one is a potential gradient. Now we shall move to another very important phenomenon, that is transport phenomenon. Phenomenon singular, phenomena plural. Now here we have in the body generally ions are present in different concentrations. So what this geochemical effect it is saying, the flow of, that is the migration of ions from one region to the another region, it is occurring. It is a gradual, it is a continuous process. It is a continuous process. For having that continuity, there must be a gradient, whether it is a concentration gradient or it is a potential gradient. So, those ions or charges must be subjected for transportation from one region to the another region. So, we have two varieties of transportation. One is passive transportation, and another one is active transportation. Now try to understand what is the demarcation between this passive transport phenomenon, transportation, and active transportation. Passive transportation means here the movement of ions is occurring along the concentration gradient. That means the meaning is, say for example, if we take water will flow from high level to a lower level generally. Similarly, if we take heat energy, then heat will flow from a body having a higher temperature to a body having a lower temperature. Similarly, if we take air will blow from a region having a higher pressure to a region having a lower pressure. It is a natural phenomenon. In this case, no need to have energy. Energy is not required. Why? Because it is a natural process. That means energy is not required. That's why the transportation is called as a passive transportation. So, passive transportation means it is the movement of ions or charges along the concentration gradient or the potential gradient. This requires no energy for transportation. Now come to the active transportation. Actually here what is happening? It is the transportation is occurring against the concentration gradient. That means from low concentration to higher concentration, ions are moving. Similarly, from lower potential to higher potential, the charges are moving. So, for carrying out this migration, this transportation, generally it requires energy. So, in the case of living systems, we have the energy reserves as ATP, adenosine triphosphate. So, for this active transportation, we give an example, that is sodium potassium pump. This is a very, very, very important topic. Sodium potassium pump, it is an example for active transportation. Then, for understanding that sodium potassium active transportation, we need two more terms. One is, what is phosphorylation? And second one is dephosphorylation. 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 Generally, phosphorylation means it is nothing but addition of, we are adding, what we are adding? PI, that means inorganic phosphate. Phosphate means PO4 minus 3. So addition of phosphate, inorganic phosphate to the protein is called phosphorylation. Generally this requires energy. So we write like this: ATP, adenosine triphosphate. It will break down and forms adenosine diphosphate and inorganic phosphate, and it releases energy. So that the energy will carry out the remaining process, that is migration of ions. So phosphorylation means it is nothing but the addition of inorganic phosphate to the protein is called as phosphorylation. Generally it is catalyzed by a specific enzyme, that is protein kinase. Protein kinase is the enzyme that is catalyzing this phosphorylation. Now, generally, which amino acids are participating in this phosphorylation and dephosphorylation? Just remember four, that is, one is serine, one amino acid, then tyrosine, then we have threonine, and the last one, histidine. You can take one more, why? Because we have this one also, glutamine. These are all amino acids. So first of all, we must know, don't get frightened. You can easily remember if you read it twice or thrice, you can easily remember these names also. But one thing we must know, amino acids' general formula is, let me clarify first. CH4, this is called methane. Okay. Suppose if we remove one hydrogen, it will give CH3. What is its name? It is called methyl. Suppose if another hydrogen is also removed, then we have here CH2. This is called methylene. Remove one more hydrogen, then you have CH like this. Now this is called methyl. Okay. Now, in order to remember this one, CH2. So here we have the CH2. Amino acid means it is having amino group NH2. And acid, that means it is having carboxylic acid also. So both amine and carboxylic acid, both groups, functional groups are present in this carbon compound. This is the actual name is glycine. The simplest amino acid is glycine. So carbon valence it must be 4. So 2 hydrogens are replaced already. So this is the methylene. So in the third, I think the third or second semester, probably you might have heard malonic ester, acetoacetic ester. They are nothing but methylene compounds, active methylene compounds. So here, this is methylene group. On one side it is having the carboxylic acid group, and on the other side it is having the amine group. Now, if we remove this, if it is a CH2, you must call it as glycine. Suppose if I remove this H1H and if I write R, alkyl group, then it is the general formula of a amino acid. So amino acid means it is nothing but it must have amino group as well as a carboxylic acid group. Now come to this serine. It is one amino acid. Then here instead of R, it will have CH2OH. That's it. So this is called as the serine. Now instead of protein, now I will write this serine structure. That is NH2, CH, COOH, then CH2, then OH. For adding this inorganic phosphate, where it is getting added here, H must be replaced. So H will go out. So here minus H positive. Then we get NH2, CH, COOH. This we can write like this also. COOH. Then here CH2. Then we have one O. Now this phosphate, inorganic phosphate is being added. Phosphorus with one oxygen double bond, single bond oxygen is carrying negative charge. So minus 3 will become minus 2. So like this here, this is a serine phosphate. So phosphate group is attached to the protein. That's why this process is called as the phosphorylation. This reaction is catalyzed by protein kinase. Now take the reverse case. That is called as dephosphorylation. So dephosphorylation. Generally here we have protein phosphatase or protein synthase. These are the enzymes which are catalyzing the dephosphorylation process. Now here what happens? Here also we have to draw this arrow like this. Once again, ADP, adenosine diphosphate plus inorganic phosphate plus energy. So this total. Here I have written those 3. They will combine and once again form ADP, giving rise to once again protein separately. That is called as, that is phosphate group is being detached from the protein. That process is said to be dephosphorylation. So phosphorylation means addition of phosphate group to the protein. Removal of phosphate group from the protein is called as the dephosphorylation. Okay, now keeping this information in mind, we can easily learn and try to understand the sodium and potassium pump. So we have here sodium, potassium pump. It is also called sodium potassium ATPase. It is one form of protein. So sodium potassium ATP, adenosine triphosphatase, phosphatase. That is an enzyme. What is an enzyme? It is nothing but a protein. What is a protein? A group of amino acids. They will form a protein. So sodium potassium ATPase is a protein or enzyme present in the cell membrane. Cell membranes of all animals. So all animals, their cell membranes, they are provided with, they are consisting of sodium potassium ATPase protein. So this is responsible for this sodium potassium pump. So let me give what is the significance of this pump, why we should learn this sodium potassium pump. This is very, very important. Why? Because it will control the concentration of sodium and potassium ions throughout the body. From where it is controlling? It is present in the controlling center is kidney. So in the kidney, from the kidney, it is controlling the balance of sodium and the potassium ions throughout the body. Suppose if it is present in heart also, we have this sodium potassium pump. Now actually in the case of heart, it will control the blood pressure, and here we have one word that is myocytes. Myocytes means these are the actually medical term, meaning is contractile cell. A cell that is contracting. So here myocytes, these are responsible. They will contract and they will subject blood for propulsion. Propulsion means movement of blood. So why blood is flowing in the blood vessels? Why? Because the myocytes are contract and they are pumping the blood. So that is distributed in the heart cells also. Okay. Now here, for having that control, generally calcium ions are very, very, very important. That is, calcium ions, it is controlling the signals on one side. That is, electrical signals means potential gradient. That we have to consider electric potential. And on the other side, the contraction of these myocytes. This will be controlled by this sodium potassium balancing. Okay, that's why it is very, 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 very important. Then third, last and final here is, suppose if the sodium potassium pump it fails to function, then what happens? The cells will swollen and that will damage the total cell also. That means the cell volume is controlled by sodium potassium ions only. Okay. Now because of this importance, so even a patient who is having imbalance or defunctioning of sodium potassium pump, he will enter into coma first, then afterwards he may also get direct ticket. So that's why this is a very, very important aspect. So let me give this sodium potassium pump. So first point here is sodium potassium pump. It is actually a active transportation phenomenon. You know what is the meaning of this active? That means it requires energy. Why it requires energy? Because from low to high, the ions are moving or charge is moving. That's why it requires energy. So that is why it is called as an active transport system. Now I will draw the diagrams also. Just try to see please. Now here I am drawing a inverted V. Draw dotted line first. Inverted V. Now, this sodium potassium ATPase protein, it is having two subunits. One is alpha unit, another one is beta unit. Now here we can draw like this. So what this stands for? So this is extracellular fluid. So this constitutes intracellular fluid. So this is the extracellular fluid, this one top, and this one is intracellular fluid. Now we must represent the ions. Generally in the intracellular fluid, sodium concentration, it is low. So low, here I will write low sodium ion concentration and high potassium ion concentration. In the case of extracellular fluid, that is reverse. That means sodium ion concentration is high and potassium ion concentration is low. Okay. Now, this sodium potassium ATPase protein, it is having two units, subunits, that is one is alpha unit, that is a large sized one, and beta subunit is somewhat thin. And alpha subunit, it is having three cup-like structures like this. And beta, it is having only two lines. This much you have to draw. Now, in the intracellular fluid, we have these sodium ions. These sodium ions, they will enter through this opening. Here there is a opening. Though the concentration is low in the intracellular fluid, sodium ions, they are accommodated in the sites of alpha subunit. So here we have Na plus, and Na+. Now, ATP, what it will do? It will split up, undergoes decomposition, and it will form inorganic phosphate. This inorganic phosphate, it will attach to this beta subunit. As a result, what happens? And by utilizing this energy, it will close this opening. So in the next diagram, you have to draw inverted V. inverted V. Like now here cell membrane, because it is a closed one, you can draw directly straight. Here it must be open, so that's why you will draw. Now this is extracellular fluid, and here we have this is intracellular fluid. Now here EI, inorganic phosphate, is attached to that beta subunit and it is acting as a key. Then this opening will be closed. As a result, at the top, this one will open. So here we have 3 sites which are carrying sodium ions, Na+, Na+, and Na+. They will escape out like this. And at the same time, through the opening, potassium ions which are present. Potassium ions also present here. That is low concentration, low potassium ion concentration. Those 2 potassium ions, they will occupy those 2 sites. Now, in the next diagram, what we have to draw? Sodium ions, they have escaped. And potassium ions are lodged, or they are locked up in the sites of this beta subunit, which is carrying the inorganic phosphate. Now, in the next case, this is nothing but the phosphorylation. Now, in the dephosphorylation, what happens? Inorganic phosphate, that will be removed. As a result, what happens? In the opening present of the extracellular fluid region, that will be closed, and here in the intracellular region, these 2 units, they will once again reopen. As a result, what happens? Sodium ions, they will flow into the intracellular fluid. Now here we have to understand one important point. That means potassium ions concentration here it is low, and it is transferred into a region having the higher concentration. So it is called as the active transportation. Similarly, sodium ion concentration in the intracellular region is low, and it is transferred into the region of high concentration. That means it is going against the concentration gradient. That is why it is called as a active transport system. So like this is the mechanism of sodium and potassium pump that plays a vital role in the case of maintenance of or for sustaining life. So we must understand the importance of these sodium and potassium ions. If their deficiency, if the pump functions, if it stops functioning, then definitely the person will die. Okay? So this is the information, and you just go through the material I have already given, you will understand easily. If you have any doubt, you just call me please. Next we learn 2 more. Just 10 minutes, I think. 10 to 15 minutes. We have 2 important enzymes. One is carboxy peptidase. Carboxy peptidase A. And second one is carbonic anhydrase. So these are the 2 enzymes that are also there in our syllabus. Each one is an important short answer. Now let us take the carboxy peptidase. So here I will write CP. carboxy peptidase and carbonic anhydrase. That is the next step. So these two enzymes, they are zinc metal possessing enzymes. So that's what we call zinc metalloenzymes. Zinc metalloenzymes. So the center of coordination is zinc metal. It is having oxidation state +2. So this is playing a vital role in that complex. Now this carboxy peptidase, it is actually having a egg-like structure. Egg-like. Like this. So here we have zinc +2. And here it is the coordination number actually it is 4. So we have on one side it is linked with 2 histidine amino acids. Histidine. And in histidine, nitrogen atom is the donor atom. So that's why we have written. So this is histidine. N is the donor atom. On the other side also, N, and this will be histidine. And next we have here glutamine. Glutamine, another amino acid, and it is having oxygen as the donor atom, and it is giving a pair of electrons to this zinc metal. And fourth one, this is EM. Okay. Now this is the structural aspect of carboxy peptidase. Now actually the carboxy peptidase totally is having 307 amino acids, and its molecular weight is 34,600. So molecular weight is 34,600 and it is having totally 307 amino acids. In those amino acids, zinc metal must be linked with 2 histidines with nitrogen as the donor atoms and 1 glutamine whose oxygen is acting as the donor atom, and the 4th valency is vacant. Okay, now what is the importance of this one? I will give it later. Now I will write comparatively. We can easily remember. That's why first I will give information regarding carbonic anhydrase, then we will learn the functions of these two enzymes. So here carbonic anhydrase is also a zinc metalloenzyme, and this one it is having 259 amino acids. So 259 amino acids are present in this carbonic anhydrase, and its molecular weight is found to be 30,000. 30,000 is the molecular weight of carbonic anhydrase. Now its structure is, it is a tetrahedral structure. Tetrahedral means you have to draw like this. A small line, bigger line, smaller, then bigger. Then add these ends, then that shape constitutes a tetrahedral. You know this one. So here center of coordination is zinc, and remaining 4 valencies. 3 valencies are occupied by histidine. Now here we have histidine, this is histidine, and this is histidine. Now there is a slight change here. I told you that the general formula of amino acid is this one. So NH2CHR COOH. Now here this H, if we remove it, then it will have O minus. Generally, nitrogen is having a pair of electrons, lone pair. So, it is a polydental ligand. That means it can give electron pairs to the metal that is having more than one donor atom. They are called polydental ligands. So, in the case of this carboxy peptidase, histidine, in histidine amino acid, nitrogen is acting as the donor atom. Whereas in a carbonic anhydrase, it is the oxygen atom that is acting as a donor atom. So you must remember that one. So here I will write O and the connection will be his. Now oxygen is the donor atom. Similarly here also, this is histidine. And here also O with histidine. So 3 histidine amino acids with their oxygen donor atoms, it is being linked with the zinc ion. And 4th side is having water mark. Okay. Now let me give the function of this one first. Now both enzymes are very important. Why? Because first I am coming to this one, carbonic anhydrase. What is its function? Generally, our blood, which is having a pH of 7.35 to 7.45. So generally, pH is expressed in a scale that is having 0 to 7 and 7 to 14. So this one from 7 we have it as this, and from 7 to 1 this is acidic. So if the pH is less, it is called as acidic in nature. If the pH is greater than 7, then it is a basic in nature. And what about 7? This is neutral. That is neutral. That is neutral. So here pH of our blood, human blood, it is having 7.35 to 7.45. It is a slightly alkaline in its nature. So this blood whose pH, it must be maintained constant. It should be maintained constant. Otherwise what happens? Suppose if the acidity of the blood, if it is less than 7.35, then we get a defect that is called as acidosis. It is called acidosis. Suppose if the pH is greater than 7.45, then it is called as alkalosis. So alkalosis and acidosis, both are the defects. So they will damage the blood. That means the pH must be maintained constant between 7.35 to 7.45 for carrying out all the biological activities. If they want to go in a smooth way, we must maintain the blood, the pH of blood, it must be 7.35 to 7.45 only. Now, how this is possible? This is done by our carbonic anhydrase enzyme. That means here the main reaction is catalyzed is H positive plus HCO3, that is bicarbonate ion, that will give us H2CO3. Then that will be converted to H2O plus CO. Now here we have bicarbonate ion. Its concentration is regulated or controlled by the ketone. And carbon dioxide, it is under the under control of, that is lungs. Lungs will control. Suppose we have acidosis, that means pH is going below 7.35, then it will increase the concentration of bicarbonate ion. Then suppose if the blood pH is moving towards alkalosis, then we have to control the carbon dioxide. So carbon dioxide must be escaping from the lungs. So like that, here this total reaction that is catalyzed by the carbonic anhydrase. Okay. Now here remember two important points. Extracellular fluid. So in the previous diagram, sodium potassium pump, there I have written sodium ions in the extracellular fluid. It is more. That means we have NaHCO3 or H2CO3, carbonic acid. That is acting as a buffer solution. And in the case of intracellular fluid, we have potassium ions are higher concentration, so we have KHCO3 or H2CO3. Okay. Now let us try to understand what is the meaning of this buffer. So buffer action means, buffer solution means it is nothing but a weak acid. So a weak acid plus its salt. That solution is called a buffer solution. So what is the speciality of this buffer solution? It will maintain a constant pH. So here weak acid, which weak acid that is functioning in our body? Carbonic acid. H2CO3 is called as the carbonic acid, and its salt. Let us take either sodium carbonate, bicarbonate, that is HCO3, or you can take potassium bicarbonate, sodium carbonate, Na2CO3, that is another salt, then K2CO3, like this. So a weak acid and its salt, that solution constitutes a buffer solution, and the speciality or significance of this buffer solution, its main important function is it will keep up, it will sustain, it will provide a steady means a constant pH. So like that, our blood whose pH is maintained 7.35 to 7.45, that is constant. So like that, this is very, very important. That is carbonic anhydrase, that is responsible for our blood maintenance of blood pH, that is 7.35 to 7.45. Thereby it is preventing us from acidosis. Suppose if I drink daily mugs of citric acid, then our blood must become acidosis. That means the pH must fall down. Similarly, if we take suppose basic substances, if we consume, then our actually digestion means the food must enter into. The food is nothing but generally we say carbohydrates is the one component, and next one is protein, and we have next one fats. So whatever we eat, that contains mainly three major biomolecules, that is carbohydrates, then proteins, then fats. Carbohydrates, they are converted into glucose, simpler molecules, and proteins, they are converted into amino acids, and fats are converted into fatty acids. And these simple molecules, they will enter into the blood. Then we say that digestion is completed. So like that here, that means the simple molecules, they will enter into the blood. That's why the pH, there is a chance of alteration in the pH of the blood. But that is not happening. Why? Because the carbonic acid, it is saving us, okay, from acidosis and alkalosis. Okay. Now I am coming to this, that is last and final, that is carboxy peptidase. Now I have not given its function. Its important function structure is this one. That is enough for this one. Now, its function is, it is used for hydrolysis of protein. Hydrolysis of protein. How protein is formed? It is the combination of amino acids. Now I am writing here NH2, CH, 1R, then COOH. Another amino acid I am writing NH2, CH, or COOH. Plus another third one, say NH2, CH, or COOH. Now here, a long chain of these amino acids, how they are linked with one another. They are joined by means of, now, a amino acid generally is represented by this portion, carboxylic acid portion. This is called as a carbon terminal. On the other side we have nitrogen, so this is called as a nitrogen terminal. So this carboxy peptidase A, that enzyme is doing the hydrolysis of a protein, long chain protein. By hydrolysis what happens? A long chain protein will be cut into short chain protein. That means breaking of protein chain is happening. Where it is breaking, it will break only at the carbon terminal. That is the important point regarding this carboxy peptidase A. So now first I will write its protein. Now here we have OH and one hydrogen. Okay, then it will expand out in the form of water molecule. Here also one OH and one H2. So here also one water molecule. So totally there is a minus 2 H2O. Then we get here NH2. There it is not disturbed. Nitrogen terminal. Then CH. Then we have R. Then CO. Then we get directly NH. Okay. Then CH. Then R. Then CO. Then once again we get NH, CH, then R, then COOH. This is not at all disturbed. So this COOH, that is carbon terminal portion, there only it is subjected for hydrolysis, that is breaking. So we are getting a long chain like this. This is a protein. So carboxy peptidase enzyme, it is catalyzing hydrolysis of a long chain protein that will be converted into short chain proteins. Say for example, I will write like this. Say leucine, one protein. Say phenylalanine, another protein. Say glutamine, then serine, like we have taken 4 amino acids. It is a long chain, let us imagine. Now it is subject for this catalyzed by carboxy peptidase A. Now at the C terminal, carbon terminal only, it will undergo a break. Now here there is a loss of one water molecule. Okay, then we have leucine, LEU, then phenylalanine, then glutamine, plus serine, separate amino acid. Like that, the breaking of a long protein chain into shorter units is done by this carboxy peptidase A. Okay, right. Thank you very much.